Digestion and absorption. Thanks to the public holidays, it wasn't 2 weeks ago that we started this topic. It was a little bit longer than that. Okay, but if you think back, we went through the alimentary canal. So I see there is a lot of names that I recognize on the list, but just in case you weren't here for that one, what we're doing, this is a two-part theory series. Reminder, I'm your live lesson teacher. Okay, so I'm here just with you on a Monday. If you have a topic that you want for a live lesson, please don't forget to send it through on the web chat, and then we can add it into the calendar. So we've covered the digestive system, so all the different bits, how they're connected, and the major functions of each area. And now we're going to look in more detail at how digestion and absorption work. So we're looking in particular at mechanical digestion, chemical digestion, and absorption. So the idea is that not only do you know where they occur, but you know a little bit more about them. And it's really important that you actually understand the difference between these, because mechanical digestion has key things that apply to it, chemical, key things for it, and absorption, key things for it. So particularly mechanical and chemical digestion, you need to be sorted on the differences between them. So we're going to start off by going through mechanical digestion in more detail, then chemical digestion, then absorption. But I'm going to need more space than I've got here. So we're going to start off with mechanical digestion. Mechanical digestion is very simply breaking things down. Okay, so we are breaking larger pieces into smaller pieces. And this is critical. Because remember, when you break something big into lots of smaller pieces, you increase the surface area. When you increase the surface area, you have more space for enzymes to do their job. Okay, so that is then bringing in chemical digestion. Mechanical digestion is just simply breaking it up to increase the surface area. Okay, there is no chemical change. Very simply cutting it up. No chemical reactions at all. So where does this happen and how does it happen? I'm hoping that the first of these you're going to be very familiar with, even if you haven't got to this topic yet. In your mouth, you have your teeth. Okay, and your teeth cut everything up into smaller pieces. This is mechanical digestion. This is where it starts. Okay, so your big word for this one here is mastication. This is just a fancy word for saying chewing. And often people will stop here. They'll say, okay, well, that's mechanical digestion. It's not the only mechanical digestion we have. We also have mechanical digestion in your small intestine. And this is the action of bile. Okay, so bile is working on your lipids. And what it's doing is it's taking those great big fat droplets and breaking them up into smaller droplets. It's mechanical digestion. It doesn't change the fat. It just breaks it into smaller droplets, increasing surface area. The fancy word for that process is emulsification. So we say that bile emulsifies our fats. It emulsifies our lipids. So in both cases, your teeth in your mouth, your bile in your small intestine, we have mechanical digestion, very simply breaking things from big pieces into small pieces to increase the surface area with no chemical change. Okay, so I keep repeating these same three things. Breaking into smaller pieces, increasing surface area, and no chemical change. Those are your three really important parts of mechanical digestion. So because IG biology is wonderful and they want you to learn just all sorts of everything, we don't leave it at this. We actually take a little bit further when we're talking about the mouth and the teeth. So let us take a moment to digress and get to know the teeth a little bit better. What do you need to know? The teeth. Now, I am not going to draw a big fancy diagram of different types. So let's work with just a simplified version of a tooth. Because you do need to know the different layers. At the top, you have your enamel. That's the shiny white bits we can see. Underneath your enamel is your dentine. In the middle of your teeth is your pulp. And then on the outside at the bottom, we have what's called cement. Okay, that's how your teeth actually stick into your gum. So the bit that you may have come across also before is your pulp, because this is where your nerves and your blood vessels are. So when you've got pain in your tooth, it's being detected by nerves in the pulp. So you've got your pulp, you've got your dentine protecting, and you've got your enamel, is your surface on the very outside that's really doing a lot of the protection. Okay, but the thing is that we have here is, I'm sure that you've come across the concept of cavities, and I'm sure you've come across the concept of fillings, and that cavities can get quite sore. What happens there is that we start getting tooth decay, and we get nastiness that's coming in, and we're decaying the enamel, and we're decaying through the dentine, and then we get into the pulp, and then it gets very sore. So, a little bit more detail, because this is not, this is biology. We've got to be a little bit more technical, okay? So what happens if we have high sugar, and then we get formation of plaque? So plaque is sticky, it's nasty, okay, but it is also got bacteria. So plaque is bacteria in a sticky matrix. So we don't need to know what that matrix is made of, okay, we can just understand that it exists. But the important thing is that we've got bacteria there. Now, you've come across bacteria before, and you've come across sugar and bacteria, and the fact that the two of them get along quite well. So what happens is that your bacteria use the sugar. Okay? We have respiration. So they're going to respire the sugar in your high sugar diet, and this produces acid. And what happens when we produce acid? Acid and enamel don't get on well together. So the acid then dissolves the enamel. When it works its way all the way through the enamel with our red scribbling, then it's going to dissolve the dentine. And through all of this, no real difference in your life. But I tell you, when it hits the pulp, hey now, That's gonna hurt. Oops, if you can spell pulp, it helps. Okay, and also now you've got bacteria and you've got your blood vessels and you've got soft tissues, and so what you get in here now is you can actually get further infections, which is going to get even more sore. So in understanding your teeth, we're linking here to diet. We've talked about the effects of a high sugary diet and all of those things that are nasty in the world that have bad effects on general health and well-being, but they also have a bad effect on your teeth. So it doesn't matter which teeth we're talking about. We can be talking about your incisors. We can be talking about your canines. We can be talking about your premolars or about your molars. They all have the same basic structure that we've got here, and therefore they've all got the same basic effect of high sugar leading to bacteria, leading to respiration, leading to acid and the dissolving of the enamel and creating of nasty pain. Okay, so one thing I do want to touch on here is that in some other syllabi and some other courses, you might have come across a need to learn tooth formulas. So there's this many incisors, this many canines, premolars, and molars. At IG Biology, you don't go into that level of detail as you're learning. Okay, if you come across it in a question, then they will give you the information that you would need for a dental formula. What you need to know is your basic function of each of these teeth types and how this links into mechanical digestion, like breaking down of the food from big pieces into smaller pieces to increase surface area with no chemical change. And that is where we're pulling in our teeth. So, enough of talking about no chemical change. Let us now actually move on to how that chemical change happens. This means we are now moving on to chemical digestion. Here we are bringing into account different foods. We're bringing into account biological molecules. We're working with enzymes. So it's another one of those lovely topics where everything comes together and where it points out that it's really important for you to know your stuff as you go. Don't wait to the end and revise, learn it as you go. So chemical digestion is all about chemical change. It's about changing large, insoluble molecules to small, soluble molecules. So the thing is, you know, we've got all this food inside our digestive system, but we have to be able to get it out and into our bodies in order to be able to use it. So those large molecules, the starches and proteins, we can't get them out of the digestive system. So we've got to break them down into something smaller, which we can then get into our body, which we can then take everywhere else, which we're going to talk about in absorption. So I'm not going to digress. So highlights of what we're talking about here. We are talking about starch being digested to maltose, which is then digested to glucose. We are talking about amino acid, back to front, proteins being digested to polypeptides, which are then digested to amino acids. And we are talking about lipids being digested to fatty acids and glycerol. So you see, it is a lovely revision of biological molecules, because when you do biological molecules, you learn that glucose molecules join together to form starch. So digestion is taking starch all the way back to glucose. And the same thing for proteins and lipids. Amino acids going to proteins, proteins back to amino acids. Fatty acids and glycerol making lipids or fats, and lipids being digested down. So we're taking everything down to its components so that we can have absorption. So we're going to look at each of these in a little bit more detail, because we need to know what enzymes are involved and where things are happening. We'll start off with starch. So we have starch going to maltose, which then goes to glucose. Starch to maltose happens in two places. And we have salivary amylase. That should give you a good clue of where it is. Salivary amylase is in the mouth. And we have pancreatic amylase. So pancreatic comes from the pancreas. So you should know where this is happening as well. After talking about the system itself, this is happening now in the duodenum. Because remember, that's where all the fluids from the pancreas come out into the duodenum. That's where they join our digestive tract. So we have salivary amylase and we have pancreatic amylase. Both of those are breaking down starch to maltose. So we've gone from lots and lots and lots of glucose to only 2 glucoses stuck together. A really funky thing here, if you take a slice of bread, when you eat bread, it's not really sweet. Don't swallow that piece of bread, just chew and chew and chew and chew and chew and chew and chew for a long time, okay? Because that happens, you give that time for that salivary amylase to digest in your mouth. So you end up not having starch in your mouth, but having maltose in your mouth. And what you'll find is, of course, maltose is a sugar. It tastes sweet. So before you swallow, your bread is going to turn sweet. So you can actually do an enzyme experiment in your own body. Okay, so anyway, we've got to maltose, but maltose has to get to glucose before we have absorption, and we have maltase. So ASE, often remember an indication we are working with an enzyme. They like to end enzyme names in ASE. Where does this happen? This happens in our ileum. So duodenum, ileum, both part of the small intestine. Go back and revise that if you need to. Remember, we said it's important to be very familiar with all the terms so that you know where things are happening once we get started talking about them. So maltase is our enzyme. Maltose will be our substrate. Glucose is our product. And now we have starch all the way down to glucose in various different places. So next one will be proteins. Proteins, we said first go to polypeptides. So we break them up a little bit, and then we break them up completely to amino acids. Our first enzyme involved with proteins is our very special enzyme, pepsin. Pepsin is our special one because he's the one in the stomach. So pepsin works at really low pHs because we've got high hydrochloric acid in the stomach. Once we get to our polypeptides, polypeptides leave the stomach into the small intestine, in particular the duodenum. So the next phase happens in the duodenum, and that is catalyzed by trypsin. So proteins to polypeptides by pepsin in the stomach, polypeptides to amino acids by trypsin in the duodenum. It is important that you take the time to learn this. It's one of those factual pieces of knowledge that you need to be familiar with, and you need to be happy with all of the terms in order to be able to take it further. So yes, you could be asked for straightforward regurgitation of this knowledge, or you could be asked to use it in a question. Something I just wanted to point out now here, in your textbook, there is reference to peptidase in your ileum as well, but that's not part of your syllabus. You've got to bear in mind that because you're covering so many different topics at IG, you're covering them all only to a certain depth. So we don't get into all the detail of every enzyme that is involved in digestion. Okay, so what you need to know for your IG Bio is proteins to polypeptides to amino acids, and we've got pepsin and we've got trypsin. So if they bring in another enzyme, okay, let's say enzyme X is working on the digestion of polypeptides in the duodenum, and they could ask you something about enzyme X, and you would have to use your knowledge of pepsin and trypsin to make an educated guess as to what's going on. For example, they could ask you, at what pH does enzyme X work? Well, if enzyme X is working with trypsin in the duodenum, the pH is going to be close to the optimum pH for trypsin. It's not going to be an acidic pH, because pepsin has our acidic pH optimum because it's working in the stomach with lots of hydrochloric acid. When all that chyme leaves the stomach, one of the things coming in to help the next set of processes is your bile. So we're neutralizing all those acids from the stomach. So we're then working at a more neutral pH. Right, so after proteins, we're going to talk now about lipids. Lipids, we only get one arrow, straight to fatty acids plus glycerol. So our lipids are our odd ones out. Our enzyme is lipase, and it's happening in the, you guessed it, duodenum. There's a lot going on in our duodenums. Okay, so remember also that lipase was an odd one out before. Sorry, not lipase, lipids. Because we talked about mechanical digestion for the lipids as well. So mechanical digestion of our lipids, we have big droplets into small droplets. We're increasing the surface area, so lipase can happen. When we increase surface area, this reaction can happen much more efficiently. Efficiency is always brilliant when it comes to biological processes. So there you have them. Starch, proteins, lipids, all the way to glucose, amino acids, fatty acids, and glycerol. All of the locations, all of the enzymes, make sure you know them. What happens next? Glucose, amino acids, fatty acids, and glycerol all have to get into your body. The next process is absorption. So this happens in your ileum. Predominant. Remember, we had all of that digestion going on in the duodenum. So now we're into the ileum, the second portion of your small intestine, and all the absorption is happening. Okay, also we have that breakdown of maltose to glucose in the ileum. Prime site for absorption. So in our ileum, we have a whole lot of villi. Okay, so your ileum wall actually looks like this. And we have increasing surface area. As soon as we increase surface area, we have more action on our processes, in this case, for absorption. Okay, in our villi, we have blood vessels. In particular, we have capillaries. Okay, these bring the blood close to the surface. So we have a very short distance to get our molecules from our digestive tract into our capillaries. And the capillaries, they take the glucose and amino acids. You also have lacteals. And these bring your lymph close to the surface. Why is this important? Because the bulk of your fatty acids and glycerol are transported by your lacteals. So everything moves across the epithelium into either capillaries or lacteals, and then it can be taken all around the body to wherever it's needed. And that's what assimilation is. Assimilation happens within your body cells. It's how the cells themselves then take in these glucose, amino acids, fatty acids, and glycerol and build it into part of the cell or use it for processes. For example, your glucose is taken into the cells and it can be stored as glycogen, or it can be used in respiration to provide energy for processes going on in the cell. Your amino acids are taken into the cell and they are built into new proteins. Remember, we can't build amino acids, we have to take them in and then we can build them into new proteins. So if we need new enzymes, for example, we can build them from the amino acids. And that's again why diet is so important, because if you don't have an intake of protein, you can't digest your amino acids, then you can't build new proteins. So if your body wants to build new proteins, you have to eat proteins. So you can digest down to amino acids and then build up into new proteins assimilation. Fatty acids and glycerol, your lipids, are used to build cell membranes. So they are vitally important. If you think about processes such as cell division, we wouldn't be able to have new cells if we didn't have new membranes. Okay, so glucose, amino acids, fatty acids, and glycerol all build bigger molecules. Digestion is that process, mechanical and chemical, that take those bigger molecules from our food, break it down into smaller molecules, which we can then take into our body and use for the things that we eat.